Good morning, everyone, and thank you for coming to Date with a Cure. For those of you I haven't met yet, I'm Eugenia Welch, the president of Alzheimer San Diego. Today, you'll have the special opportunity to hear about the latest in Alzheimer's research directly from the scientists working to find a cure. We're also fortunate to be joined by a researcher who specializes in caregiver stress, which is such an important topic for all of us here today. On that note, I would like to recognize our esteemed panel for being a part of this event, especially for being flexible to spend their Saturday morning on Zoom. I also want to thank our generous partners for sponsoring us this morning. Because of their continued support, all of the services we provide are at zero cost to the community. You should have received a link to our online exhibitor scavenger hunt. It was included in the email that provided the link to join this webinar. We'll also share the link again at the end of the program if you missed it. If you haven't already, take a few minutes this weekend to complete the scavenger hunt. Once submitted, you'll automatically be entered to win a $50 Amazon gift card, and you'll have until 11:59 p.m. Sunday evening to do it. It's really quite fun, and you'll learn a lot about our sponsors. Now I want to tell you a little bit about our organization. Alzheimer San Diego is a local nonprofit. Our mission is helping people impacted by dementia, fighting stigma, and supporting research. To that end, every dollar donated to Alzheimer San Diego stays here in San Diego County to help the community we know and love. As you can imagine, there have been some changes in how we deliver our services because of COVID-19. Last year, we moved all of our programs online and even added some new ones. But I'm excited to say that as of this month, we are officially back to in-person social activities and outings. These will be held outdoors following social distancing guidelines. I hope to see some of your faces in person soon. Of course, people living with dementia and their care partners can still get the help they need from our dementia experts. Our clinical care coaches provide supportive counseling using video calls, the old-fashioned telephone, and online live chat. Our education team has done a tremendous job creating online classes. We now have a substantial webinar library on topics ranging from Dementia 101 to communication skills, preparing for the late stage, and more. While we do plan to resume some in-person classes this summer, we will continue to offer online options as well. We have dozens of Zoom support groups to choose from, available 6 days a week with morning, afternoon, and evening options. Vitals is a new program we created during the pandemic, specifically to support the 25% of San Diegans living with dementia who also live alone. Volunteers check in on them with weekly phone calls. These calls can be purely social, but they can also provide reminders about things like upcoming appointments, food deliveries, or identify a new need the person with dementia may have. Our Alz Companions Respite program is back to in-person visits as well. Specially trained volunteers spend time with people living with dementia so care partners can take a much needed break. We're still continuing a virtual version of this program for anyone who might not be comfortable having a volunteer in their home. If you need help setting up or troubleshooting any technology like Zoom or FaceTime, our volunteer tech team is here to help. All of our programs are provided free of charge to the community, thanks to the generosity of our donors. And finally, we continue to support local research by matching people with clinical trials and providing grants through our Collaboration for Cure program, which we like to call C4C. We'll revisit C4C in just a moment, but first I want to share some important news. This time last year, we knew there were 84,000 San Diegans living with Alzheimer's or another dementia. In a new report released by the county, that number has increased by almost 20%. Today, I'm sad to say there are nearly 100,000 San Diegans living with this heartbreaking disease. I want you to think about that for a moment and really take it in. 100,000 people and 100,000 local families in the caregiving trenches right at this moment. And unfortunately, we know there are many more people struggling who've not officially been diagnosed. I have an unusual goal as CEO. As much as I love my job and the powerful work we do, my deepest hope is that someday we can shut our doors. that there won't need to be an Alzheimer's San Diego or an Alzheimer's anywhere because there's no longer an Alzheimer's crisis. As you'll find out from our experts this morning, we're closer than ever to finding a cure. But until that day comes, it is an honor to serve our community and support the 100,000 San Diegans living with this terrible disease. As I've mentioned, all of the services we provide are 100% free of charge. We are completely supported by the generosity of our community, which is so critical as we work to support more people than ever before. Two of our biggest fundraisers are coming up: our annual motorcycle ride, Rides for Alls, and of course our annual walk, the Walk for Alls, which we've held for years at beautiful Balboa Park. While I know motorcycle riding might not be for everyone, who doesn't love a day in the park? And I'm hoping all of you here today will sign up for the Walk for Alls. It's free to register. And if you do like to ride, Rides for Alls is less than a month away. All bikes are welcome, and it's really a lot of fun. You can learn more about both events at the links in the chat field. In addition to our brilliant researchers, we have another special guest this morning, our longtime friend and supporter, Darlene Marcos Shiley. It is because of her innovative vision and generosity that our C4C initiative has led to some incredible research breakthroughs. And for her, this cause is deeply personal. Mrs. Shiley, the floor is yours.
I'm ready to take on the floor. Thanks very much, Eugenia. I'm not on the floor, everyone. I'm just taking it over here. I really am so proud of the accomplishments of the local research community here in San Diego County. And I've been around a long time, about 40 years involved with this particular topic. And I have to say it's been gratifying, very much so, to watch our investment in C4C yield such powerful results. I think many of you know my family involvement with Alzheimer's. I lost an uncle, an aunt, and my mother to Alzheimer's. So Donald and I, my late husband Donald and I, wanted to do something to improve the research and the clinical care and caregiver, because I learned personally just exactly what that's like. So early on, we joined with UCSD to, I guess you would say, create the Shiley Marcos Alzheimer's Disease Research Center. And I'm very proud of that organization. I hope to see it do even more over the years. Let me talk about it. Eugenia asked me to talk a little bit about the history of C4C and why it was important to me. And I think obviously the family involvement explains some things to you, but there's also, there's the need to, when you have an idea, to make something happen, you need an idea, you need a leader to promote it, and you need donors. Former supervisor, chair of the Board of Supervisors, Diane Jacob, led us in the C4C, and I matched the county funds to start and to sustain the project to this point. Eugenie also asked me to tell you a little bit about the key accomplishments of the C4C. Since its inception in 2015, over 17 research projects have been funded, and through C4C, more than 1.1 million has been awarded to support local dementia research. That's 1.1 specifically for C4C, in addition to all the other funding that goes on. And 4 of those projects were so promising, they've gone on to receive 8.76 million in funding from the National Institute on Aging. That's a pretty decent return on investment, as many of you who dabble in investments are aware of. And frankly, we're confident that even more C4C projects will go on to receive national funding. You're going to hear some updates from a few of those researchers this morning, some I worked very closely with. And something that Eugenia said that I'd like you to really remember: Alzheimer's San Diego is a local and independent organization. All the money raised here stays here. That's very important to those of us who contribute. You don't want to see your money go out and not come back. So this is an important thing to me, and I think to many. So I would ask all of you, not necessarily verbally at the moment, but to join me in thanking Eugenia and her team for bringing these researchers together today. They're extraordinary people doing extraordinary work. I look forward to hearing from them, and I think then we should get started. Thanks so much.
Thank you, Mrs. Shiley, so much. And again, we can't thank you enough for all of your support and what you have done to really make C4C what it is today. We are very grateful. And now it is my great pleasure to introduce today's moderator, Amy Abrams. She is sure to be a familiar face to those of you who have attended our community education classes or webinars. Amy has dedicated her life to helping people living with dementia and their care partners, and we are so very lucky to have her as the Director of Education at Alzheimer's San Diego. Amy, you are now in charge.
Thank you. And the luck is all mine. I'm really happy to be here. Thank you for the warm welcome, Eugenia, for your inspiring comments, Mrs. Shiley. Before we get underway, I just want to kind of go through a couple of housekeeping items about Zoom here. We're in Zoom webinar, so everyone is muted, and that's just to reduce background noise and distractions, but we very much hope that you will ask questions here today and participate with us. So can I ask Jean and our tech team volunteers to come on camera so I can introduce you here to help us keep track of questions, which by the way, will be monitored and handled by Jean Alton, who's a member of our education department here at Alzheimer's San Diego. Hi, Jean.
Good morning, everybody.
To help Jean keep all of your questions organized, we will ask you to please submit those using the Q&A function of the Zoom webinar. This is in your control panel. When you click it, it'll pop up a box that looks like this. So when you have a question for one of the researchers, please type it in, click send, and then we'll be able to see your questions and we can answer as many of them. We'll ask the researchers to answer as many of them as we can. We'll do our best. There's probably going to be a lot here today. Depending on what type of device you're using or how big your screen or your monitor is, this box might get in your way and keep you from being able to see the presenters or the slides. So just click that minimize button and it will get out of your way. So please do keep your questions to the Q&A, but know that there's also a chat function, and we will be sending out, we already have been, we will continue to send out information and materials in the chat as we go along. All of those things, you can keep them open and keep an eye on them, or you can just move them out of your view if you find them distracting. There is closed captioning available. We run a live transcript of this program. So if that would be helpful to you in following along, then just click the CC live transcript button and it will appear. You can control some of the features there. Alternatively, if it's on and you don't want it, if you're finding it distracting, you can use that same button to turn them off. Okay, quick note that this program is being recorded right now. Only we, the panelists, are being recorded, not the chat or the Q&A. So be aware of that. We will post a link to this recording when it is available in the coming days. And finally, if you're having any technical difficulty during the course of the webinar here today, please do not get frustrated. We have two members of our volunteer tech team here with us today. Tanvi and David are with us and they are available to help you if you're struggling with the video, the audio, the settings, getting that closed captioning on and off, whatever it might be. If you need some support, don't get frustrated. You can put a message in the chat. They will be keeping an eye on chat and they can follow up with you, or just call the office. 858-492-4400 is our main number, and you can be connected to one of our volunteers who can talk you through whatever technical assistance you need. So my thanks to our volunteers for being here with us today. As Eugenia mentioned, I have spent my career working with people living with dementia and their care partners. So that is to say, I know how important this event is. It's important to me, and I know how important it is to you, how many questions you have. I know what you're reading out there, what you're seeing in the news. I'm sure that the recent FDA decision on aducanumab has been a hot topic for all of us, and we'll certainly be discussing it here this morning. It can be really hard to separate the facts from the fiction and the misunderstandings. I'm delighted to give you this chance to get your questions answered directly by the experts in the field. The researchers on this panel are truly extraordinary. They are the reason that a cure for dementia could very well be found right here in San Diego County. So if you've attended this event in the past, we're going to be doing things a little bit differently this year in order to maximize the time for Q&A together. So rather than asking each researcher to give a presentation on their area of expertise, we've put together a handout with all of their bios, and we'll be covering as much of that during the course of the program today as we can. A link to that handout has just been entered into the chat. You can click it, pull that up, and follow along as we go. So to get us started, I'm just going to cover a few quick facts about dementia to lay out the issues of what we're talking about here today. As Eugenia shared earlier, there's about 100,000 San Diegans now living with dementia. 98,610 is the exact tally, but we know that many more cases go undiagnosed. It is the third leading cause of death here in our county, and the numbers are only expected to grow in the coming years. Just a quick note that when we refer to statistics about dementia, we are not just talking about Alzheimer's disease. Much of the research that's going to be described here today is focused on that specific type of dementia. It is the most common, but we're engaged in the search for a cure for all of the underlying causes, including Lewy body dementia, vascular cognitive impairment, the frontotemporal disorders, and many more. A great deal of research has been done and continues in identifying the risk factors for Alzheimer's disease and other dementias. We know that age, sex, and health conditions such as hypertension, heart disease, diabetes, and untreated hearing loss contribute to a person's individual risk, as does one's family history, education levels, and other factors. At this time, there are no known environmental causes of Alzheimer's disease. Important to note that income and health disparities do contribute dramatically to higher rates of dementia that we see in communities of color in the United States. And there's often very poor representation of Black, Latinx, Asian American, and Indigenous people in clinical research, something I know that members of today's panel are working to address. But while the cure still eludes us, research continues to demonstrate that the best thing you can do to keep your brain healthy is to keep your heart healthy and focus on your overall health. Eat a nutritious diet, stay active, engage in intellectually challenging activities, and I assure you today will be an intellectually challenging activity. Get social and focus on getting good quality sleep. All of that's a lot easier said than done, I know. Living with dementia as a person affected by the symptoms or caring for someone who is a tremendous challenge. We know that the needs are many and that they are significant. Meeting these needs, this is what we're here for as an organization. We'll remind you again at the end of the program today how to get in touch with us if you're in need of support. But in the meantime, just know that we're here to help. An extensive amount of information, resources, referral to community supports, and of course, much needed emotional support. Supporting local research efforts and ensuring that our community knows about opportunities to participate in those research efforts is a key part of our mission. So as Mrs. Shiley mentioned, we do want to highlight today some of the successes that have been made possible by our Collaboration for Cure, or C4C initiative. It's my pleasure now to introduce Dr. Robert Rissman from the University of California at San Diego, whose research concerning stress hormones and Alzheimer's disease received $1.73 million from the NIA last year. Dr. Rissman, welcome.
Thank you. Good morning, everyone. Thank you, Eugenia and Amy, for inviting me here today. I want to talk to you a little bit about a project that I had initially funded through the C4C program that, as Amy just mentioned, was able to move on into a major NIH award. So as she mentioned, I'm professor of neurosciences at UC San Diego. And part of my research involves trying to understand the role of stress and the role of stress receptors in Alzheimer's disease and how drugs that impact the system can be used as treatment. Next slide. Okay, great. So just as a very fast overview of the stress system. So the main stress peptide in your brain is called corticotropin releasing factor. And this hormone actually initiates the stress response in your body, prepares your body for stress. It interacts with two receptors, two main receptors, CRF-R1 and CRF-R2. And that's how all of the functions of your body are coordinated when CRF is released. So how do we think that CRF is involved in Alzheimer's disease? Well, there's been a lot of research over the years that have looked into CRF. And it's very clear that CRF changes occur in Alzheimer's disease. There's actually less CRF in your brain as Alzheimer's disease progresses. This occurs very much in vulnerable areas to Alzheimer's pathology, including its presence around beta amyloid plaques in the brains of Alzheimer patients. And there's actually changes in the affinity of the whole stress system, of the whole CRF system, in terms of its ability to bind as Alzheimer's disease progresses. Next slide, please. So my lab has been involved for many years in looking at the impact of CRF and also the impact of agents that block CRF, called CRF receptor antagonists, and how those can potentially help with Alzheimer progression. We started with animal models, and we've found that indeed the drugs that do exist to block CRF do help a lot in reducing not only the cognitive impairments that these mouse models develop, but also the neuropathology that they develop. The problem has been is that there aren't any viable CRF receptor antagonists that can be used in humans. Many of them were developed over the years, but they're simply not safe enough or not effective enough to be administered to a human. So what I proposed to the C4C program is that we change the whole outlook of looking at CRF receptor antagonists. And instead of looking at the back part of the receptor, let's look at the front part of the receptor where CRF actually binds. And so that was funded through the C4C program. And I have worked very closely with the people at Sanford Burnham to generate a scaffolding and drug development program to be able to develop new CRF receptor antagonists focused on the N-terminus, the front part of the receptor. That went very well. And as I mentioned earlier, we applied for an NIH grant to expand it and to really prosecute the compounds that we found and determine which ones would be most effective. And that's what we're doing right now in our NIH award. So the program is going absolutely very, very well. I was really very, very thankful to receive the C4C award. It really started this program off when, really the NIH at this point was not interested in this. This was an old subject, and they were like, we have other things that we really prioritize. But the C4C program saw that as a possibility to really help this program, and it made a massive difference. So thank you.
Thank you, Dr. Rissman. The story is very inspirational, super inspiring. Great to hear the direct outcome of the C4C project like that. Thank you so much. So next, we'll hear from Dr. Gerald Chun with the Sanford Burnham Prebys Medical Discovery Institute. He'll be sharing some exciting developments about two of their research projects. In addition to some of his own research, he will also be telling you about some of the work of Dr. Elena Pasquale. She couldn't join us today, but her project was also supported by C4C. Welcome, Dr. Chun.
Thank you, Amy. And hi, everyone. I hope you've passed through this pandemic safely. Be really looking forward to times when we can just get together in person. And thanks, of course, to Eugenia. And it's great to see Darlene Shiley's face there. We've missed you. And you're looking good. And really, all of our colleagues here are actively working on different aspects of what we'll be talking about today. And the C4C came from some really early discussions that Darlene and, as you heard, Diane Jacob had to bring some seed funding into the discovery of possible new agents that might lead to drugs in the future. And as you heard from Robert, it's really hard to get things off the ground. So C4C has been integral in developing some of the first steps for that. And so I'll take you through 3 little programs that are still at that seed stage, but where we think we might be able to make some inroads. One additional comment, actually, if we go back to that slide, Amy. One additional comment to make is that I'm talking about what in the industry you refer to as small molecules. So the drugs you get in a pill, for example. And that's in contrast to what you'll hear later about antibody therapies or biological molecules, which is a separate class of molecular agents. So what this particular program will look at here is illustrated firstly by these little Tinker Toy colored pictures to the left in this first slide. And those are little chunks of genes that are being brought together to create new little genes. And that process appears to involve a specific enzyme called reverse transcriptase. And now if I could go to the next slide. So reverse transcriptases can be inhibited. And so this C4C project is to really look at new ways of inhibiting the reverse transcriptases by creating small molecules. And the concept is that in Alzheimer's disease, you can create those little molecules from pathogenic genes that I just mentioned, and they have a little name called gen cDNA or genomic cDNA. That's not really important here today, but that process involves this reverse transcriptase reaction as shown in the colored diagram sort of towards the bottom of this from a Khan Academy slide. So you all know we start off with DNA, and that DNA then goes to RNA, and that RNA then goes to the protein. And the reverse transcriptase reaction takes the RNA and sends it back to DNA. And so that process appears to be occurring within the brain, and it appears to involve enzymes that are endogenous, that belong in the brain and get dysregulated somehow in Alzheimer's disease. So if we could block some of those processes, maybe we could find some new ways of reducing the incidence or the severity of Alzheimer's disease. So that is one project that we're undergoing, trying to find some new small molecules that can block these reverse transcriptases. And if we now move to the second slide, yeah, we can just pass through that. Yep. It's a completely different project, but one that's built on also a clinical literature looking at some lipid receptors. And so lipids, of course, are very important in the brain. They're the most common element of the brain by dry weight. And there's a huge literature on the importance of lipids, both in, of course, our normal bodies, where they're talking about cardiovascular fitness. As you heard, the heart and brain are intimately linked. And of course, the gene ApoE, which is a lipid binding protein or complex. And so this is a kind of lipid that's called sphingosine 1-phosphate or S1P. And in fact, some of you may have come across this if you know anyone with multiple sclerosis. So this particular receptor class, which came from actually studies that we had initiated 25 years ago today, identified the first of this family of receptors. It turns out that those are now the targets for some 4 FDA-approved multiple sclerosis medicines. And it may be that some of the same lessons we've learned from multiple sclerosis might apply to Alzheimer's disease therapeutics. And so what this particular receptor can do is it can reduce the amount of exocytotic glutamate. Those are those gray balls there. If you have too much glutamate in your system, it can damage your cells. And so the idea is that we might be able to make small molecules to inhibit this particular receptor and therefore reduce the exocytotic glutamate responses. So that's just an example of a second project. And then the third project, which is one being driven by Elena Pasquale. So in the next slide, she's looking at what are called Ephrin-4 or A4 Ephrin receptors. And these are illustrated in this nice diagram. It has that nickname FA4 receptor. It's a single transmembrane domain receptor. That is, it crosses into the cell and goes from the outside inside with one single pass. And it may be that some of these receptors can bind and respond to peptides from A beta that are in Alzheimer's disease. And so the idea is to create inhibitors of this FA4 receptor, and those studies are also being supported by C4C. And so with that, I'll stop and hand the microphone back to Amy. Thanks for your attention.
Thank you so much, Dr. Chun. It's very exciting research. And again, we're so proud of all that we've accomplished through C4C. Here at Alzheimer's San Diego, we are a local nonprofit organization. So we're really excited to be able to welcome this this panel of local experts to hear not only about C4C funded research, but about the broader advances that are happening in research here. So remember, this is your chance to get your questions answered directly. If you haven't already, find that Q&A box and go ahead and submit those questions. We're keeping an eye on them, and we're gonna get going with our panel here. Now, can I ask all of our panelists to turn on cameras and let's say hello. Okay, hopefully you can see everyone here. Dr. Paul Asen is the director of USC's Alzheimer's Therapeutic Research Institute. Dr. Jim Brewer directs the Shiley Marcos Alzheimer's Disease Research Center at UCSD and is the chair of the UCSD Department of Neurosciences. Dr. Brent Mosbach is a licensed clinical psychologist, runs the Alzheimer's Caregiver Study at UC San Diego, identifying strategies for helping patients and caregivers minimize stress. Dr. Michael Plopper is the medical director of Sharp Mesa Vista's Clinical Research Center, where he leads several clinical trials about evaluating new Alzheimer's treatments. And Dr. Sherry Safji is the co-founder, CFO, and medical director of Excel Research, which partners with major pharmaceutical sponsors to conduct clinical research trials for new medications and treatments of several conditions, including Alzheimer's disease. So I think with that, you have now met everyone. So let's get to some questions. I'm going to pull down the slides. And we'll get going here. Okay, I'm going to start with you, Dr. Asen, if I may. Actually, here's what I'll do. If I can, I'll sort of open this up to whoever wants to take it first. Before we get going with the questions that have come in through the Q&A, I'd love just a, sort of an overview of what is happening around the FDA's recent decision around aducanumab, because I know that this is front and center on everyone's minds right now. Who would like to take on the task of providing us with an overview of the treatment, of its approval, of its status, and what it means to our folks who are living with dementia today? Great, Dr. Asen, you're on.
Yeah, I'm happy to talk about this. It's a tremendous milestone for all of us in the field. This is the first approval of a disease-slowing medication for the treatment of Alzheimer's disease. We've been working on this for decades. It is an enormously important event. At the same time, it's a very complicated one. And so I appreciate the opportunity to talk for just a minute about it. The drug is aducanumab, and aducanumab is an antibody. It's an antibody that attacks amyloid deposits. Amyloid deposits occur in the brain. They are a hallmark, the pathological hallmark of Alzheimer's disease. Many of us believe that the disease starts with the accumulation of this abnormal material, the amyloid. And so many of our efforts have focused on amyloid as a therapeutic target. And aducanumab is an antibody that binds to these deposits of amyloid in the brain and actually promotes clearance. That is, it stimulates brain cells called microglia to attack the amyloid and actually remove it from the brain. So essentially, this approach to treatment allows us to target the primary abnormality and get rid of it. Sounds great. We've been trying to do this for 2 decades without success. And aducanumab is the closest, well, now approved amyloid targeting therapeutic. Why is this controversial then? Isn't that just great? Well, having a great idea and a great candidate treatment is not enough. There are many details that have to be addressed. How do we deliver the treatment? At what dose? How do we select people to get the treatment? How do we measure the benefits of the treatment? What are the side effects of the treatment? And there are complex answers to all of those questions. Let me just say that aducanumab has managed to have a beneficial effect in one of two major clinical trials that led to a submission to the FDA. Aducanumab clearly removes, even normalizes amyloid in the brain, and when administered to people at a specific stage of disease called early AD, which is mild dementia and prodromal AD, then not only does it remove the amyloid, but it appears to modestly slow the cognitive decline in Alzheimer's disease. So it's not just changing the presence of this lesion, the plaque, the amyloid plaque, but it does seem to slow progression. But it's been difficult to prove that. And there were two large trials. One was positive and one was negative. And that's the source of much of the controversy. Normally, the FDA would require two positive large studies before approving a drug. So why did the FDA approve aducanumab when one of the large studies was negative and only one was positive? Well, in part, it has to do with the enormous need to have something that will actually slow disease progression. And it has to do with very detailed analysis of all the data that explains, at least in part, why one of the trials was negative. And the FDA, after much debate and continuing debate in the field, has decided that the benefits outweigh the risks. But there are risks. Removing amyloid from the brain is associated with changes to the small vessels in the brain as they try to deal with this load of material being removed. And that has toxic effects, including small bleeding events in the brain called microhemorrhages and areas of swelling in the brain. These changes occur in a significant number of people who are treated with aducanumab. And sometimes they cause symptoms. Often they don't. Often they're mild. Sometimes they cause symptoms. Sometimes they even require treatment. And so there are risks as well as these benefits. And that's the reason for all the debate. What does it mean, Amy, you asked, what does it mean for people with Alzheimer's disease? Well, it means there's an option now. It's not cut and dried. There are risks. There appear to be benefits in a certain segment of the Alzheimer's disease population, but it is now available. Aducanumab is available for clinical use. It's a biologic that has to be infused intravenously every month, and it's expensive, and we're still waiting to hear whether payers like Medicare and private insurance will cover it. I suspect that they will, but probably under specific circumstances only. So for people with Alzheimer's disease, there's now an important topic of discussion with your clinicians. Are you someone who would fit the characteristics of people that seem to respond to this? You need to understand the potential risks and think about and discuss with your clinician the benefits and risks of this treatment. But we do now have a clinically available therapy to slow down progression of Alzheimer's.
Great, thank you. That is very helpful and actually covers a lot of the questions that I saw coming in through the Q&A. So thank you for that really helpful overview. Dr. Plopper, if I could just ask you, what does it mean to say that a drug is clinically available? And does that mean that it's just available to folks who are participating in research? Could you explain that further?
Well, welcome everyone, and nice to see you all virtually. That's going to be the big question here: is how will this new medication and future medications be disseminated to the public? Dr. Azen mentioned payers. Well, actually, if Medicare were to approve payment for this, and the volume of activity were to be as great as one could easily project, Medicare would be bankrupted. The amount of money spent, it's anticipated on this particular drug, could be as much as all Part D spending currently. So this is obviously an important factor, which may turn out to be very much a rate limiting step. The other thing that is important with this is that this very likely will depend on biomarker availability and positivity. I mean, you don't want to give an amyloid reducing drug to someone who doesn't have amyloid. So in all likelihood, there will be a PET scan required. And Dr. Rissman's work with biomarkers may lead to a plasma detected alternative, which would make things a lot less expensive and easier to carry out and would have a lot of ramifications in terms of this drug. But it very much remains to be seen on how this will be disseminated. The drug was sent to by Biogen, probably predominantly the clinical trialists who did the trials. I think there were 600 sites, and they are sending the drug now. So they're going to start disseminating the drug through those sites initially. But we probably don't have enough specialists to be able to actually assess people well enough and deliver this drug well enough right now. So this will require probably more specialists to help us to administer this. As was mentioned, I believe one nice outcome of this, I think, will be that more people will seek diagnosis. We currently don't do a great job in general practice in terms of detecting dementia and helping people with the early stages of dementia. And so this, I think, will promote a lot of activity relative to diagnosis and just general awareness.
Great, thank you. Very helpful, and I imagine more questions on this topic are going to come up as we go along, but really appreciate that insight. I have a question for you here, Dr. Brewer. What is the research community doing to improve the diversity of participants in clinical trials right now?
Oh, great. I like that question. That's excellent because it's a it's a place where we at the Shiley Marcos Alzheimer's Disease Research Center really put our bets on for this coming cycle of renewal from the NIH. So in addition to Darlene's very generous donation to us that helps get us going and pursue innovative approaches that are high risk, high reward, we also obtain $15 million every cycle from the NIH to help push forward our work. And one of the really missing aspects across the nation's Alzheimer's program is the understanding of diverse communities, and in particular, where we can really fit in and help is in Latino communities. And so what we did this cycle is put forth a whole new core that focuses on Latino recruitment and understanding Latino science as it relates to Alzheimer's disease. We in fact have started a partnership with San Diego State University and the South Bay Latino Research Center in Chula Vista to actually do work in the community to make it much easier for our Latino partners to come to our center and get the full evaluation in their community. And we're very, very excited about that. We're actually ready to launch our inaugural visit where we would do the entire, what we call the uniform data set collection, including blood and other markers in Chula Vista in our partner site. It's very exciting. Why is this so important? It's key because so much of our work is dependent on studies done in European Caucasian participants, and particularly the genetic basis of Alzheimer's disease. What we're seeing with one of our new recruits, Hector Gonzalez, who runs the largest MCI and Alzheimer's disease cohort across the nation, is that ApoE4, one of the main risk factors we've talked about many times in the past and is always in the headlines as the main genetic risk factor that predisposes you to Alzheimer's disease, may have very different impacts for risk in the Latino community. In fact, it may not be the most relevant marker. And so all this work that's going on to treat ApoE4 may not be as relevant to diverse communities. And that's why I think it's so critical to recruit individuals and get participation from diverse communities into our studies. One of the things we also do that I think is super exciting, we stayed true to our neurosciences roots by starting what's called an induced pluripotent stem cell core. Well, what does that mean? Well, it means that the new human models of the disease can be made from skin biopsies from our patients. You can actually turn those skin biopsies into neurons and other brain cells in a dish, so you can understand the impacts of genetics and the molecular pathways that were never really clearly laid out in the animal models. Animal models were critical to get us to some point, but really they're missing some human aspects of the disease. So if you merge our recruitment of Latino subjects with things like disease in a dish models, it means places like Mayo Clinic, Harvard, other places that don't have access to Latinos can still do science relevant to Latinos. So because we can share those samples with others. So very, very excited about that.
Things that are in the chat box.
Terrific.
Hey, David,
Sorry.
our tech
Awesome.
team
Okay,
volunteered.
you
Mute
can
yourself,
copy and
David.
paste. You
Great. Thank you. The perils of Zoom and webinar technology. Thank you so much. That's very exciting developments there all around. I certainly hope that we at Alzheimer's San Diego can be supportive in those efforts in the future. On the topic of looking for broader subjects for the field, the various things that we're studying when it comes to Alzheimer's and other types of dementia. Dr. Mosbach, I'd like to ask a question of you. We are interested not only in finding a cure for this disease, but in finding ways to support people who are affected by the disease, emotionally, socially, physically. Can you give us just an overview of major interventions that are currently being studied with regard to caregiver health and well-being?
Sure, absolutely. I appreciate the opportunity to answer the question. And what's happened over the last 20 years is that we've actually found that there are a number of interventions that are extremely helpful for caregivers. We have a number of evidence-based interventions. I think there has been more than a dozen that seem to help reduce distress in caregivers. The issue has become getting access to the caregivers, so giving them access to these interventions and making them available. So the most recent push has been for taking these evidence-based interventions and using technology, either through things like Zoom or telemedicine or app-based interventions that teach basic evidence-based skills to caregivers through an app, and then they can apply those and monitor how well those particular skills are working. So that has been a very big push from the NIH to try and develop modifications to existing evidence-based interventions, primarily that are based on cognitive behavior therapy, which examines how caregivers perceive events, the kinds of thoughts they have. Are they using things like, oh, this always happens, it's never going to ever get better? I mean, you're using this all or nothing kind of thinking, and trying to help people sort of broaden and step back from that instead of looking down a telescope just at the negative kinds of things that are going on. So that's really what's been happening, is taking evidence-based interventions primarily based on cognitive and behavioral kinds of strategies and making them available to caregivers either through technology or through telemedicine kinds of formats.
Great. And what would you say to a care partner who is interested in participating in some of this research?
Well, for lack of self-promotion here, I mean, of course, I'll be doing that. We have a program that's going on right now for caregivers at UC, and I can provide more information on that as well. It's actually an app-based intervention that's designed to help people use specific coping strategies to improve their well-being. But in addition to that, there's other kinds of places that you can look. The National Clinical Trials, clinicaltrials.gov is the website where you can go on and do a search for keywords like caregiver, and you can find studies all over the country that are enrolling caregivers. And Research Match is another website that people can register at, and people can find not only caregiver-based interventions, but other kinds of clinical trials that are based on for their partners who, or family members or loved ones who are dealing with the Alzheimer's disease or related dementia. So those are some key areas where people can look to find existing either clinical trials or programs that might be available to them.
Great. Thank you so much. I hope folks will avail themselves of those opportunities. And just a reminder that we do maintain on our website, on the research section of our website at Alzheimer's San Diego, information about local opportunities to participate in various types of research. So thank you so much for paying attention to the well-being of our caregivers as well. Dr. Sofji, this is probably kind of a hard question, but just in broad strokes, what is required of someone who wants to participate in one of the trials happening there at your organization, or in research more broadly?
Sure. I'll try to keep it as simple as possible. The main things to keep in mind is that at any given time, there are many different trials going on, and a site like mine or Dr. Plopper's may only be doing one or two of those trials. So educating yourself about what's going on is important. The sites should help you sort of figure that out to some extent. And each trial has certain criteria that people have to meet. Again, we would help decide that. So the first step is if someone sees an ad or finds out about a trial they're interested in, they would call someone like my site or Dr. Plopper's site or at UCSD, and we would help educate you about that, as well as figure out if you might potentially qualify. Given that at times the person with the disease may not necessarily be the person calling, it is important that the person with the disease be on board and have an interest in that. We can't, you know, we want people wanting to do research, not being pushed into it by caregivers. So anyway, that phone call starts everything, and then we would take it from there. And essentially we walk you through the process where we figure out if it's a good fit for you, give you all the information you need to decide if you think it's a good fit for you. And then there's many different steps, you know, through the testing. You know, for a drug like aducanumab, obviously you had to have a PET scan that was positive for amyloid plaques as a qualifying factor, and each study has something like that. So there's many different steps along the way, but we keep the people participating and their caregivers informed at each step and let you know kind of why we're doing what we're doing and kind of what it's going to mean for you in the study participation. The actual study requires that you attend visits and go through different procedures. And again, each study is different. And we've all adapted during the pandemic as well. So sometimes it didn't require in-person visits as they used to, but most of the time it does. Things like cognitive testing do not lend themselves well to Zoom or telemedicine. So anyway, the participation in a trial is typically at least 18 months. So it is a long commitment. But again, it can be a very worthwhile endeavor, either to potentially get a new medication or just to contribute to the development of a cure or some other, you know, a treatment for Alzheimer's or their dementia. I did want to put a plug in. There are several trials going on for what is officially labeled agitation in Alzheimer's or agitation associated with Alzheimer's. I just want to make the point that agitation is sort of overstated. That is a term that I believe the FDA came up with, but it really refers to a lot of the distressing behaviors that occur in the moderate to severe Alzheimer's or middle to late stages, and which can be very disruptive for the life of the person with the disorder as well as their caregiver. And so until we do find a cure or better treatments to prevent people getting to those stages, we do need some sort of treatments that will help people get through those difficult stages of the disorder. And so those are the other kind of trial that are going on. Although, of course, you know, the curative or preventative or, you know, things that ameliorate the disease are such, you know, much more exciting and obviously things that we're all wanting to try to get on board with first.
Yes. Thank you so much for that clarification about that language and that it originated at the FDA. That's very interesting. I hadn't heard that before. Because yes, we are seeing and reading a lot about those studies right now. So thank you very much. I have a question that I'm not quite sure who to pose this to. This might be Dr. Chun or Dr. Rissman or Mrs. Shiley. This one might even be for you. I don't know. It's just an interesting question. And since you're still here, maybe I can put you on the spot. But somebody has asked if there are research initiatives in other areas of the country that are funded similarly to C4C. This collaboration that we see happening here locally in San Diego County is very exciting. It seems maybe like it's rare, maybe it's not. What type of collaboration? Are there other programs funded like this around the country? Does anyone have any insight?
Since you tried to plug that one on me, Amy, I'm going to
Great.
take a shot at this and
Great.
say, I have no idea.
Mm.
But if there is, I'm sure that ours is the best because there's no question in my mind about that. I really don't know. When we first started this at that time, that was 2015. I'm not aware that there was anything at that time like this. But obviously, with our incredible results, I mean, really to invest 1.1 million and in this short a period of time, everyone knows how hard it is to get to the national agencies. I mean, it's well, I won't, since this is being taped and they're already auditing me for my excessive charitable contributions, I don't want to give them anything else to work with. At that time, there wasn't anything. I would guess that there probably isn't now, but I'm sure that one of our illustrious doctors here can give you much better information than I just spurt out there.
That's great. Well, you did really well for just being thrown on the spot. So thank you very much. And again, we're all so grateful for your having gotten a project like this started here in San Diego County. So proud of it. Dr. Risman, do you have any insight there?
Sure. I agree with Mrs. Shiley that this is a very unique program, very unique to Alzheimer's San Diego. As I speak to my colleagues, nationally, they think it's a great program, but simply do not have it at their local Alzheimer, either Alzheimer, whatever their city is, or Alzheimer's Association. I'm not sure why. It's really a great program where we can fund local science and really, give seed funds to things that wouldn't be funded otherwise. So, yeah, my answer is I don't think so.
Okay. Okay. So we are as special as we think we are. Yeah.
Yes, you are very special. Yeah.
Okay, great. Well,
Let me add that. No,
yeah, please.
no, there's. Oh, sorry, Amy. No,
Please, go
yeah,
ahead.
no, there really is a specialness to this, and it relates to the community that is so interactive. So that my colleagues across the Mesa, whether it's Jim at UCSD or Robert at UCSD, or folks that are at, say, the Salk Institute or Scripps, or we have that access. And then secondly, that's not widely known. There's a drug screening system that's required to get at a number of these types of small molecule agents. And the Prebys Center at Sanford Burnham Prebys is probably the best academic drug screening center in the country. It's certainly up there as one of the best, and it really rivals what you find in industry. So having access to that, tying it into C4C makes it a very powerful system that can actually deliver. It's not just words. So I would second the notion that this really is a unique resource and something very special here in San Diego.
Great, thank you. Well, we'll just continue to be very proud of it and look for more innovation coming out of it. Dr. Asen, I believe this is a good question for you. Are there other, sort of what else is happening in drug development? Are there other disease modifying drugs on the horizon that are close to getting FDA review, possible approval? Where are we?
Yeah, so there are thankfully many, many programs underway at all phases of drug development. I think that the drugs that are closest to possible approval are most often in a similar class to aducanumab. They're anti-amyloid interventions. And I saw one question in the Q&A about Lilly's Trailblazer study, which was just announced a few months ago. And that is a positive study of another antibody that targets amyloid and reduces amyloid in brain. That drug is called donanemab. And in the positive study, which was not a phase 3, but a phase 2 study, treatment with donanemab normalized brain amyloid and slowed progression as measured by cognitive and functional assessments. And phase 3 studies are now in progress for donanemab, and they're also in progress for two other similar amyloid removing antibodies. And those are lecanemab, also called BAN2401, and gantenerumab. 4 different antibodies that are generally like aducanumab that are at late stage, phase 3 stage of development, and are likely to be reviewed by the FDA in the coming years. There are also many other efforts, and some involve earlier intervention with antibodies, trying to get the amyloid out even before there are symptoms. Others target the other main lesions. So we've talked about amyloid, but there are also the tangles made up of a different protein called tau. And there are many antibodies and other therapies that are targeting tau that are now in phase 2 studies. There are a large number of studies that target the inflammatory component of Alzheimer's disease, the activity of cells like microglial cells in the brain that seem to be important, and a whole slew of other targets. So in answer to your question, I think it's great news that there's a lot of research going on around amyloid, around tau, and other targets. Some will be potentially reviewable by the FDA in the coming few years. Others will take longer, but coming years are going to show a major advance in slowing down Alzheimer's disease.
And can I build on that? There was a question in the Q&A about Mark Tushinsky's innovative program, and this is another great collaboration that we had. Dr. Azen had run the original, very innovative approach of doing injection of nerve growth factor, and now Dr. Mark Tushinsky is launching a new program with brain-derived neurotrophic factor in a similar vein. These are very innovative, high risk, high reward approaches. And it's one of these areas where we see our clinical participants, the clinical trial volunteers, as the heroes of this fight. They are the ones that give their comfort and take risks on our behalf to advance this field. And so just noting that that's a neurosurgery trial. These are probably one of the most aggressive approaches where you're actually injecting into the brain itself to try to help bolster the brain's survival mechanisms, and to have brave patients and caregivers participating in such a trial, it's really what allows us to move forward in this fight. So I wanted to bring that up.
Thank you very much for acknowledging the courage and the bravery of those participants, and actually takes me to a question for you, Dr. Plopper, if I may. Are there opportunities, this is kind of a two-parter, are there opportunities for people who are not diagnosed, who are healthy, not care partners specifically, but people who are interested in participating in Alzheimer's and other types of dementia research? That's the first part. And the second part is, how do you navigate, to Dr. Brewer's point, those questions of, medical interventions that are going to happen, informed consent around for people who have declining decision-making capacity? How does that work?
Well, as to the first question, Dr. Azen is sponsoring through USC and ATRI a study of lecanemab in preclinical Alzheimer's. So these are people who may have a family history, have a strong interest in participating for one reason or another, and will present themselves for a PET scan, which would determine whether or not they have amyloid pathology at a certain level to warrant participation. And we also do genetic testing for those folks. And if they don't qualify, then we have a disclosure meeting with them in which we reveal to them that thankfully they don't have amyloid in their brain. And then we reveal to them their genetic testing and the results of that. So this is in preclinical Alzheimer's disease. It's amazing the amount of interest in participation, and especially for people who don't have any symptoms. So this involves either twice monthly or monthly infusions, depending on the amount of amyloid in your brain. And so it's and for a lengthy duration of time. So it's not a small endeavor in any respect in terms of volunteering for this and putting up with this and participating in this. One of the nice things people are finding, though, through this screening process is whether or not they have amyloid. So just participating in that level, getting a PET scan, which is awfully hard to get in private practice, that can determine this, makes it worthwhile for people just to find that out. And then if they do have amyloid, to participate. But it's that's an example of a preclinical Alzheimer's state, in which maybe Dr. Easman will want to comment further on that. And then the second question had to do with medical interventions. Would you remind me of that question?
Sure,
again?
it was just sort of a general question prompted by Dr. Brewer's description of a particular study. Just a question about how do you navigate the trickiness of subjects providing informed consent when there is the possibility, the eventuality of losing decision-making capacity in a person who's living with dementia?
Well, at this point, most of the studies that have been described and that we're doing are for folks who are very early in the course or don't have symptoms. So that's a pretty easy call to make, that they are able to give informed consent. But as the disease progresses, and sometimes people present for other types of studies with a more significant dementia evident, and it either would be that the person is capable of giving informed consent, and I would do that assessment, or have a legally authorized representative, and there are specific definitions of that within California law as to who would be capable of signing for the subject. And so in those instances, we have the subject sign and the legally authorized representative. So that is a pathway for involvement for people who have questionable informed consent capability. The other thing is to be able to reconsent people along the way. I mean, it's we have to mention these are placebo-controlled trials, and generally half the people are potentially receiving a placebo. They may worsen over some years involvement in a trial, and we may need to reconsent them as to their capacity to continue in the trial at that point. So that's part of the process.
Great, thank you. Yes, Dr. Asen.
Yeah, well, thank you, Michael, for talking about the AHEAD study and letting people know about this intervention in asymptomatic people. I wanted to mention, it's okay to self-promote for a second, another part of this program, which is called the APT web study. And anybody can join the APT web study if they are at least 50 years of age. That's when we start to worry about brain changes that could be linked to Alzheimer's. And to find it, it's just aptwebstudy.org. And the web study just takes 15 or 20 minutes every 3 months. We monitor your brain health. We share with you the findings that we have. We share with you information about clinical trials such as AHEAD. And if we see evidence of increasing risk, we establish a connection between you and one of the sites for an early intervention study. So we would love to have anybody who's interested or worried about their brain health join the APT web study.
Terrific, thank you. I have a question that's come in specifically for you, Dr. Rissman. Can you speak to the link, the relationship between Alzheimer's disease and Parkinson's disease?
Yeah, I thought that was a very interesting question. You know, when I took over the Shiley Marcos ADRC neuropathology core in 2015, it was as a basic science researcher who really thought Alzheimer's disease was a very pure disease, right? In our animal models, it certainly is. When we talk about it, it very often is. In our trials, you know, we recruit Alzheimer's subjects. And then, you know, we proceeded to do about 40 or 50 autopsies that year, and nearly all of them were what you would call comorbid. Almost everyone had alpha-synuclein pathology. Some of them had TDP 43 pathology, in addition to amyloid and tau. So I think it's probably more common than we think it is to have AD and PD, particularly in later stages of Alzheimer's disease. So I think it's a very interesting question. Perhaps it's almost a different subtype of the disease as it becomes very mature.
Great. Thank you for.
I'll just
clarifying.
add
Please.
that the comorbidities are dramatic, and there are extensive studies not only from Robert but from many different groups that have defined these comorbidities, including vascular dementias, in addition to the Lewy body types. So comorbidity is more likely the rule than the exception, and that's something that all of us will have to keep in mind as we look at these disease etiologies.
Right. So to that end, I have 2 large NIH grants that study exactly that. Alzheimer's disease with Parkinson's disease, and DLB and Alzheimer's disease, also dementia with Lewy bodies and Alzheimer's disease. So I agree, it's a very major area. I don't know how common it is though earlier on in the disease progression though. And I don't know if others have comments on the thoughts on that.
How common the known comorbidity.
To have these combined, either combined symptoms or combined neuropathology in early stages of the disease. Again, to my experience in our neuropathology core, some of our earlier patients, as they're earlier in disease, if they pass away, those are more pure cases.
Anyone have a comment or an observation on that?
I guess I would point out that even in young people with genetically determined early onset AD, 50% have Parkinson's changes, Lewy bodies in brain. So that the two, to me, that's very strong evidence that the two are linked. So even though we're talking about a disease that typically starts in the 40s, when there are Alzheimer's changes, half the time there are associated Parkinson's.
While we're on this topic of comorbidities and sort of expanding the definition of what we mean by dementia, Dr. Mosbach, can I ask your study or other caregiver intervention studies that you know of, are they looking beyond the Alzheimer's caregiver and looking at people who are caring for people with Parkinson's disease, frontotemporal dementias, these other issues?
Oh, absolutely. I think it's actually the exception that to clinical trials or any studies where people are exclusively limited to Alzheimer's disease. So the tendency of almost every clinical trial that I'm aware of through NIH or other mechanism is that this is Alzheimer's disease or related dementias. I would think that if you tried to get into a clinical trial and you know that your loved one has Parkinson's or Lewy body or any other kind of dementia, you should be eligible for those trials.
Okay, great.
Because
Thank
the
you.
issue really is that the caregiver is in some level of stress or distress, and they can get that through any of the dementias, not just Alzheimer's.
Right. Great. This is a really wonderful question. I do not know who to address this to. So let's say, who among you went to medical school the most recently? With all of the panelists, the panel's tremendous work and efforts, I'm anxious to see that the importance of this research is translated into action by practicing physicians and neurologists. How much emphasis is given to dementia in required ongoing training for practicing physicians and neurologists? Dr. Brewer, can you address
Yeah, I
that
should
one?
jump in on that as a
Great.
kind of a chair of the department, wearing my chair of the department role and the education of our medical students role. At UC San Diego, it's big because we are an area that this has been one of the feathers in the cap at this center. I mean, under the leadership all the way back to Bob Katzman, Bob Terry, Leon Thal, even George Glenner, who basically made the connection between amyloid and Alzheimer's disease. That percolates through to our trainees. I can't speak to many of the other centers, other places I've been, Stanford and Johns Hopkins, not as much as at UC San Diego. Another reason why it's really important that, the work of Alzheimer's San Diego really is building on the awareness in our local community, not just diffusing it across the entire country. This is a very special place, I have to say. And so at our center, yes, we are a lot of our grand rounds. We have a neurodegeneration seminar series that is in place at the Shiley Marcos Alzheimer's Disease Research Center. Every 2 weeks, we have a esteemed speaker, and now with Zoom, it's actually even easier. We have international speakers. We have just the best of the whole area, and even when we're out of the pandemic, people like to come to San Diego, so we get a lot of educational opportunities. So our trainees, our medical students, our neuropsychologists, and our graduate students get a very healthy infusion of training on the latest and greatest that is happening, not just in San Diego, but worldwide. I will say one of the really nice things that's happened at UC San Diego is the training grant opportunities that have happened. So the work that has taken place to bolster funding from the NIH for Alzheimer's funding also led to training grants that we've secured. Our center has, our center alone has 3 training grants that are focused on Alzheimer's disease. So we are always looking for trainees to educate them. We have a new, what's called a research education component within the Shiley Marcos ADRC, where we give each of the trainees $35,000 a year to pursue a project that will actually get their feet wet in the science and deliver and just under, with the little training wheels on. We have a great mentorship team to really guide them along in their science, but we give real money behind not just education, but actual real money to pursue a project. And I think it's really exciting to do that. We also have, again, with this recruitment of Hector Gonzalez, who was coming from Michigan. Very easy to recruit the guy because he says, get me out of Michigan, get me into San Diego, and especially in a place where there's a lot of Latino subjects that he could work with. He has a cohort of over 4,500 Latinos in the South Bay, and he helped us get this what's called an Alzheimer's Disease Resource Center for Minority Aging Research. So we're one of 5 across the nation to have received specialized funding to bolster science of underrepresented, and in our case, focused on Latinos. So yes, a lot of education to our trainees, and it's bolstered by the help of Darlene Shiley through the funding of our program, other philanthropists who dedicated funding to what we call the Emerging Scholars Fund, that we were able to actually give dollars to not just say, hey, here's a training, here's an education, here's a lecture you should go to with your busy day, but here's actually some money to put behind this and actually get you started. Very excited about what we're doing in training.
Terrific. That is very, very encouraging news. And again, may what is happening here in San Diego spread elsewhere as well into other medical training programs. I'll just make since we're open to making plugs, I'll just promote the County of San Diego's Alzheimer's project is very invested, the clinical roundtable in particular. And we have several representatives from Alzheimer's San Diego who participate in the clinical roundtable, working on ways to improve general primary care providers' capacity to support patients who are living with dementia and their caregivers. So I'm very proud of that as well. Dr. Safji, we often, being the director of education, I field questions on pretty much a daily basis about cannabis, the use of THC and cannabis in Alzheimer's treatment, caregiver stress treatments. Are there any of the pharmaceutical companies that sponsor the type of research studies that your company is conducting? Are they working in the area of cannabinoids? Are you seeing anything happening there?
Well, I'd like to open that up to the other people. They may know more than I do, to be honest. Generally, no, I have not really seen that. And unfortunately, frequently, it is still something like you can't come in and test positive for cannabis and enter a clinical trial for most of the trials, which I personally disagree with. But
oh, so that's an actual barrier to participating in the trial. Okay.
Yes, not always, but many times.
Thank you. Dr. Asen, did you have something to add?
Yeah, we actually are working to start a study of cannabinoids for the treatment of Alzheimer's disease. The idea behind the study is a focus on end-stage dementia, where agitated behavior and other behavioral manifestations are so prevalent and so disturbing. So this study, which is funded by NIH, will enroll people at the latest stage of dementia who have behavioral issues, and it's a randomized trial of cannabinoid therapy to see if we can gain a benefit, to see what the risks are in this population, and really to inform the potential use of cannabinoids in the management.
Great. I'm sure many of our folks in attendance will be happy to hear that. We get that question often, and very rarely have anything to share on that front. So thank you. I have a question for you, Dr. Mosbach. Advice for people who are working in long-term care, healthcare, as well as family caregivers, sort of practical strategies based on your research for avoiding burnout.
Oh, great question. Well, I'll give a very simple one that I've been working a lot with lately. It's an acronym, right? GRAPES. And each of the letters in the acronym stands for something we can do on a daily basis that'll help us ease some of the distress that's associated with caregiving or whatever other kinds of stresses we're dealing with. So G is you have to learn to be gentle with yourself. Don't talk. Don't use negative language to yourself. Try to avoid negative languages about the situation you're in, because it only adds to the suffering. R is find ways to relax, deep breathing, mindfulness, meditation, yoga, or anything that's personal to you that helps you get a sense of relaxation. You have to incorporate that into your daily life. A is we have to find ways to get little accomplishments and take them and accept them as real. And so set up specific goals you have for yourself each day. And sometimes those goals can be, I want to wake up today and I want to compliment 5 different people, or I want to be kind or do something nice for someone. And then you can count how many times as you go along what you do. P is you have to incorporate pleasant activities into your daily routine, because they provide us with a sense of meaningful activities that are good for you. They provide us with a sense of pleasure and joy, and they are uplifting. Sometimes we lose touch with those things because we're so focused in on the other kinds of stressors that we're involved with. E is exercise, and S is to socialize with positive people. So I've been giving that acronym to a lot of people, and they seem to really want to run with it because it's simple. It's straightforward. You can put a little thing on your refrigerator or next to your bedside, and then you can wake up and say, okay, what am I going to do in each of these areas every day? I hope that's helpful. So
Yes, hugely helpful. Thank you. Here's another question. I'm not totally sure who should answer it. I'm going to throw it at you, Dr. Chun, and you let me know. Just bounce it around if it needs to go elsewhere. What role, if any, should SPECT scanning have in diagnosis?
Yeah, that's out of my realm.
Okay.
And Paul or
Who
Jim
wants it?
Falls into my wheelhouse.
Okay,
So
thank you.
before I took my major administrative roles of my big thing was biomarkers and imaging biomarkers. And in fact, my first exposure to Alzheimer's disease was with Bill Jagist at Lawrence Berkeley Laboratory, doing not PET scans. PET scans are the better version of SPECT scans, but we were doing SPECT scans in the very first iteration of this. And it does give you a sense of the hypometabolism or reduced blood flow in areas of the brain that are affected by Alzheimer's disease. It is not our preferred approach. We've moved far beyond SPECT scans in our diagnostic process of imaging.
Okay.
Just want to add one thing about that, Amy. Diagnosis this week is tremendously important. It was less important last week. This week we have a treatment, aducanumab, that's directed at a specific pathology in brain, amyloid, that should be measured before considering a treatment for it. And the standard way to measure amyloid is using amyloid PET scanning. And that's fine, and it allows us to really visualize the plaques and decide whether someone might benefit from a treatment like aducanumab. But it's very expensive, and at least as of now, it's not reimbursed. It typically costs about $7,000 to get a scan. But I wanted to mention that we are getting very close to the full validation of blood tests for amyloid in brain. Those tests seem to be very accurate, and before long, we will be using a blood test to show whether someone has amyloid in brain, and it will likely be useful to select people for treatments like aducanumab.
Terrific. I was hoping that was going to come up today with all of this news about aducanumab. It seemed like the need for the availability of those blood tests is urgent. Yeah, great. Thank you. I have a kind of a juicy question here, and I don't know who wants to take it. So I'll let you all decide. But why did the 3 members of the FDA committee quit in protest of this week's decision?
Well, I'd be happy to.
Okay, great. Thank
To
you.
address that. I spoke about this briefly at the outset. It was a difficult decision. Typically, the FDA requires 2 positive phase 3 trials to approve a drug. And it's particularly important when a drug carries risk, as aducanumab does, and when it's enormously expensive, as aducanumab is. So the FDA decision to approve aducanumab with 1 positive phase 3 study and 1 negative phase 3 study is very controversial. And at the FDA, 2 different teams were involved in reviewing the application and came up with opposite conclusions. The clinical team strongly supported approval, and the statistical team strongly opposed approval. So how do you manage that? The advisory committee sided with the statisticians and voted against approval. The FDA Division of Neurology decided that the totality of evidence, particularly since it's now supported by positive evidence from other similar drugs like donanemab, meant that we should bring this as an opportunity to the millions of people who have Alzheimer's disease, and they granted what's called accelerated approval. And that means it's approval, the drug can be marketed, but additional information is required by the FDA, such as another clinical trial, and that the approval can be withdrawn if there is not supportive information from another clinical trial. However, the question, why did 3 people resign from the advisory committee? The advisory committee considered carefully the wealth of information, saw the statistical concerns with 1 positive and 1 negative study, voted 10-0 with 1 abstention against supporting aducanumab, and yet the FDA has provided an accelerated approval. That's pretty frustrating. That's why 3 members of the committee have now resigned.
Yes,
Amy,
Dr. Chen.
Yeah, so speaking from the science side, I think the scientific community was quite puzzled by the decision based upon 20 years of both kind of failed clinical trials and ambiguity in the scientific literature. In fact, there's a whole section of scientific literature that argues against these plaques being, say, causative, and they consider them as, say, scars of some other process. So it's a more complex situation, as Paul has already alluded to. And so that, I think, kind of compounded the frustration of some of the advisory committee members. It's not simply looking at the failed trial and then the partially successful trial, which was actually not so straightforward because of the high doses used in 1 arm of it. I think it was the totality of the experience thus far where many different approaches have failed. And now suddenly 1 section within 1 did seem to work. And there, of course, the data support that. And yet everything else doesn't. And so that is where the puzzling aspects come in. And we'll just have to see if there is a clear efficacy signal as we go along. And as Paul has already mentioned, there's also the safety signals that we'll have to see if they raise any issues. But bottom line is it's approved. And
yeah,
right.
So Gerald and I joined Scott Peters' town hall recently, and this was a very hot topic of Congressman Scott Peters, and the impacts to Medicare that Dr. Plopper mentioned, and all kinds of things. There's many impacts. One of the pieces also in that 1 section of 1 arm is also the group that tends to get the brain imaging signal of ARIA, this amyloid-related imaging abnormalities, which brings in a concept of difficulty in blinding and other impacts that are just so hard to grapple with, and how that might have actually influenced end results. But in the end, what I have been telling my team members who are either distraught or just trying to figure out what way we go forward, because it's not only on the research side where we're going to have amyloid negative individuals progressing. I mean, we know that the amyloid negative individuals will continue to progress, because even in the study where there was a modest benefit, people were still moving toward dementia despite the amyloid removal. But what I see as a positive is, this is going to remove amyloid from part of this component of the question that's been confusing the scientists for so long. And in some ways, I see the field needed a kick in the pants like this. And I think this is what it will happen. The field will say, geez, why are these people still progressing and I can't detect really any amyloid in their brain? So now we'll look at this tau cascade. We'll actually look at the progress that happens in the, despite the removal of amyloid. It'll be a pretty interesting field going forward. And that's where I see it as something that is going to move the field forward regardless. We do have major concerns though about how we deliver it. At UC San Diego, we're already a place where a lot of people come for their dementia care. And we just sent up a multidisciplinary Center for Brain Health and Memory Disorders. This is one of those places where you may not want to plug it because we're already booked out way too far for my preferences. If you call up and try to get an appointment with us, we're so backed up, and now the aducanumab is going to cause even more of a backup. I did use the opportunity to get 5 extra parking spaces at our center just to say, let's make use of this opportunity to get better resources. And we are asking for another nurse supported for our center. We got a nurse practitioner now, social work, neuropsychology, neuropsychometry, geriatric psych, geriatrician, and our neurologists in place. And I've been just pushing them to open up more slots, more new slots, and make use of the nurse practitioner to open up slots. But this is the impact that happened to us. We are worried, but we're excited to deliver care, and we have our infusion center ready to go for this. And it's going to be a new world.
That's great. Thank you. As the social worker in the room here, I'll express my thanks for making room for those services as well. Dr. Chen, I'm gonna pose this to you. Dr. Brewer's comments about the kick in the pants and the look at tau cascade, and this just makes me. I'm wondering if you can tell us about other theories that abound with regard to the origins, the causes of Alzheimer's disease. Any summary or update you can provide there?
Yeah, there's such a universe of different explanations, and you've already heard many of them mentioned, from the A beta theories to what extends from A beta, the tau hypotheses, which I should add are difficult to really address in, say, an animal model, because in fact, animals don't get AD, they don't get the same pathology. And even when you engineer them, they don't get the same types of phenomena. And so we try to model them in, say, stem cell or iPSCs, as you heard from Jim. But even there, those are more like fetuses or babies, and babies and fetuses don't get Alzheimer's disease. So there's a whole bunch of things that have raised a whole another class of possible things going on, one of which I alluded to and looking for at C4C, that there are these changes within the single cells of your brain that are taking place. And this is referred to as genomic mosaicism, and it can involve a recombination of different types of genes within that brain. Is that relevant? There may be some signals that are appearing. One of the things it could do is, for example, help to explain the comorbidities, that you have these processes that act on multiple sets of genes. As you already heard from Robert, you have Parkinson's types of pathology, you have vascular diseases, and yet those gene sets are totally different than what happens in the, say, familial forms of Alzheimer's disease that involve, say, APP and tau, those 2 genes. So I think there's still a very, very rich and interesting landscape that is there to be understood. And part of that relates to how much do we really understand about the brain? How is it possible for us to be here talking to each other and thinking about things and contemplating a number of other issues at the same time? These are still mysteries out there. I should say San Diego is the hotbed for neuroscience in general and trying to understand many of these aspects. So I think there's great opportunity here. And this community, all of us here sitting here in front of you, is poised to make those types of discoveries. And those will come, I think, in the not too distant future. How the therapies will manifest, we still don't know. We think there may be some other pathways, but we need the clinical trials that Paul's group's running and others here in San Diego. It's really important that these are also brought to bear, and I'm hopeful that we'll see more of that in the near future.
Great, thank you. That's exciting. I was hoping you'd touch on mosaicism. That's always very interesting topic. Dr. Safji, I wonder if you could just explain for anyone who's not clear the difference between sort of how Excel and companies like Excel work, how the trials are funded and structured compared to studies in these academic settings.
Sure, I'd be happy to. So Excel Research is an independent clinical research trial site. We're not affiliated with any medical group or other research institute. We contract with the pharmaceutical companies who are starting a clinical trial, and we agree to try to find the subjects, the people, the volunteers that are willing to participate to do those studies. Other places that are doing research get their funding from different places. They may get it from a governmental institute, from Alzheimer's San Diego, the C4C, and so their funding comes from a different place, and they may be affiliated with a larger organization, which has some impact on how they function. They may have different resources and they're connected in different ways. My company, while we can be very independent and that has some benefits, it's also, we're limited in that we're not connected with an institution like UCSD or any other great research institutions we have around San Diego. But just to be clear, some of the studies can be done at any of these research sites. So even the UCSD and Dr. Plupper's site, again, we could all be working on the same clinical trial, but my site would not be working on a trial that's only funded by, say, the NIA or another governmental institution.
Thank you. That's an important point of clarification. I know something that causes a lot of confusion, something being studied at multiple sites in the same region. Thank you. Very helpful. Dr. Plopper, I'm going to give this one to the geropsychiatrist in the room. Is there a link? What is the link between mental health, anxiety, depression, and dementia, and what is being studied in this area right now?
Well, it's a difficult question to answer, but we do know that there is some predisposition to dementia for people who have recurrent major depression in their adult lives. So there is some increased likelihood of that happening, especially if it's untreated depression. So that's, to some extent, can be a harbinger of things to come, but not always, certainly. But the most important component of this is that early on in the disorder, people with Alzheimer's disease are also are subject to other mental health symptoms and conditions. I mean, they certainly can be depression early on in the course of illness, anxiety states. As time goes on, there can be psychotic symptoms of hallucinations, and not really fixed delusions as much as just sort of amorphous changes in the way people perceive the world, but often to a delusional extent. And that can be later in the course, typically is later in the course of illness. We try and treat symptomatology early in the course of Alzheimer's with typical medications we use for these disorders outside of that, and with some benefit. But the main thing is to make sure that when we prescribe medications like that, let's say antidepressants or anxiolytics for other types of medications, that we monitor them closely, and if they are not helping particularly, that we stop them. Because what happens very often, and one of the first things we do is to de-prescribe medications when we're treating people who come into us early on, is to deselect medications they may be taking, which they no longer need to take. And that often is true for mental health oriented medications. And in particular, benzodiazepines, which can cause significant cognitive changes as well as tolerance and potential withdrawal states. So these are medicines that shouldn't be prescribed easily. They should be monitored closely, but there can be benefit if they are appropriately monitored.
Thank you. Well, well handled for a difficult question. Thank you. I know it was a big, broad topic. There's a good question here. And again, another one I'm not sure. So it's sort of grab bag. Anybody jump at it if you can. Are there any known connections between Alzheimer's disease, other types of dementia, and autism? I hear that a lot in classes that I'm teaching with family caregivers. People note the similarities in terms of some of the behaviors, as well as some of the communication strategies and behavioral interventions that are successful with people with dementia, that people say, oh gosh, this sounds a lot like how I was told to interact with my child who has autism, for example. Can anybody speak to that? Any possible connection there?
Well, I'll take a stab at it.
Great, thank you.
So autism is thought to be a developmental disorder, and many of the changes that occur early in life and perhaps during fetal life can then manifest later in different parts of the postnatal period. But all of that, of course, will involve neuronal, glial, other cell type functions or dysfunctions that can then ultimately manifest with behavioral changes that could be shared between, say, in the case of autism, what you see in children or teenagers or young adults even, versus the aged experiences that you will see in an Alzheimer's disease patient. So there's undoubtedly, there are going to be gene overlaps, just simply because there are so many shared genes that are expressed at these different periods of time, but also the types of interactions are going to be conserved. And so what we interpret as the behavioral output that's due to, say, a developmental phenomenon versus something that occurs with aging simply overlaps and involves some of the same types of genes. So yes, if we look, we will find them. However, they don't seem to be, at least at this point, the same exact genes that have been identified between the two diseases. So they are distinct, but they likely share some of the same sort of cellular and systems types of mechanisms.
Very interesting. Thank you. On the topic of genes, there's a question about how much one's genetics are a contributing factor. The question is really in the context of ethnic disparities, known ethnic disparities here in the United States, but I'd like to expand it sort of more broadly. In terms of a person's personal risk related to developing Alzheimer's or another form of dementia, sort of what's the weight of genetic factors versus other factors?
I mean, a lot of people could answer this. At least I'll speak from the component of trying to look across the genome to come up with a personalized risk score. It's something we at UCSD have been doing, and I think we have probably the best performing, what's called in our case, a polygenic hazard score, where you actually even just take the whole genome and weight the different protective versus deleterious factors in the genome. And we can get a very good sense when combining that with age at where the individual is likely to begin conversion. And we've published a few papers on this, in fact, in terms of an enrichment strategy to not use these super expensive approaches of amyloid imaging everybody, regardless of age or genetic background. You can actually save a lot of money because these, even just the GWAS that you see from 23andMe or Ancestry.com has a lot of information in it that you can weight and understand the likelihood of amyloid positivity. And then you still have to follow it up with a subset of those individuals where your bets are placed. And you will save many millions of dollars on a trial if you did it that way. Again, they are not ready for clinical practice. They're not ready for personalized use, but it's something we're studying very well. And I think the one thing that we did that other groups had not done, most of the time when people do a genome weighted polygenic risk score for a case versus control, they would say, well, let's look at a bunch of cases and then compare the genes to those who don't have the disease. Well, that doesn't work in Alzheimer's disease, because if you have a 68-year-old person who's a control who doesn't have the disease, you don't know if they're not going to get the disease in 2 to 3 years. In fact, if you wait long enough, they very much will have the pathology in their brain. So we took a completely different approach to say, let's weight the genome based on at what point you are going to get Alzheimer's disease, and just assume everybody is going to get it. But some people might get it at age 108 where it's not relevant, and some people will get it at age 72 where it is quite relevant. That really was a game changer in developing of the polygenic hazard score, and we think we've got a very powerful tool. We're, again, not yet using it in clinical practice. We use it in our research setting to understand it better. And the one piece exactly relevant to the question that came forward, well, this has been tested mostly in European Caucasian studies, individuals, and therefore our polygenic hazard score is unlikely to perform quite as well in all the diverse communities. It does still perform, we have checked it out, but we definitely need more participants from a broad range of individuals to get a better set. The one nice thing about the genome, though, is that even if not asking individual what their inheritance was, their family history, I think when you do these essentially ancestry.com type approaches, you don't even have to ask the family history. You know their history going back thousands of years just from what's embedded in the genome. And so I think that gives us a lot of power to actually model in what was inherited across the time. And a lot of times people don't know what they've actually inherited. You might have thought you were one race, and you may identify with one race, and yet the genome tells otherwise. And then it becomes very hot button issue. But in fact, the genome is the genome. And so that's what we're working at to try to get our best prediction from that data.
Wow, that's fascinating. There's something you just wouldn't have heard said 10 years ago. What an interesting innovation. Thank you for that. On the topic of clinical practice, there's a question that was submitted about Aricept. So we're talking a lot here today about the new things. Let's talk for a second about the old things. So Aricept, the question was about its efficacy over time. So I know that how effective Aricept is has long-standing, has been in argument for a long time. The question was about, this person had read that it's really only effective for a few months. Can anyone sort of give a summary of what we know about how effective Aricept is and how long it can be effective? Dr. Asen?
Yeah, I can start.
Great.
So Aricept was developed based on studies over a short period of time. The initial studies of the efficacy of Aricept in providing some memory enhancement to people with Alzheimer's disease were conducted over 12 weeks. The pivotal trials that led to approval of Aricept were 6-month studies. And the benefits are modest in size, but the benefits are certainly present over 6 months. And that, I think, has led to the misconception that Aricept only works for 6 months. In fact, there have been studies carried out to 2 years and even 3 years that show that at a group level, so each individual may have a different response, but in general, at the group level, the benefits of Aricept continue for years. And many clinicians extrapolate and believe that the modest benefits of Aricept will continue as long as you take the drug. Now, that's not to say that Aricept prevents worsening of Alzheimer's. And this also contributes to the misconception. Everybody taking Aricept declines, and they decline at a rate that's similar to people who are not taking Aricept. It is a boost in memory so that at any point in time, while you're taking the drug, your memory is a bit better than it would be without the drug, but you are still getting worse over time at a similar rate to people who are not on the drug. So a lot of confusing aspects to this, but the basic answer is it's a misconception that the modest benefits of Aricept only last a few months. They certainly last for years.
Great. That is very helpful, a very helpful point of clarification. Thank you. There's a lot of misinformation out there based on those early studies. Great. Thank you. If I may, I would like to wrap up our Q&A here by asking all of you to answer the same question. This has become something of a tradition of mine. So bear with me. And I'm just going to go in the order in which I see you on my screen. So that's what this is all about. Dr. Brewer, what thing that is happening in dementia research today makes you feel the most hopeful?
Oh, great. Yeah, so that's nice to get the first crack at this one, because there is a lot going on. What thing? Oof, that's going to be a tough one, though, because I am so excited that we're now able to study this disease in living humans, not only using the new biomarkers to actually look at progression. We now have measures of the tau deposition in the brain, as well as the amyloid deposition in the brain, and plasma markers to look within individuals. That is going to propel our science so much more than what it had in the past, where we were only reliant on genetic studies of mice that had the bad protein crammed into its brain, and it said, oh, the mouse doesn't work as well, and then you pull the bad protein out and the mouse works better, and you say, oh, I've got an Alzheimer's cure here. That's not Alzheimer's. And so feeding into that same one development is the new human cell models. And what Gerald talked about with we turn these things into fetal cells, yes, and then turn them into neurons. But actually at the Salk Institute and at UCSD, we've had direct transformation of fibroblasts into neurons, which retains the aging effect. So all of that with the genetic markers gives us a roadmap to treat this disease. We know genetic factors impact and their impacts on molecular pathways in the cell are influencing age of onset of this disease. So that gives us a roadmap to do medication development that mimics the impact of a protective gene allele. That is basically our roadmap to just say, let's start knocking these out one after another. ApoE4 has, of course, already been targeted, but there are many other factors that are protective or deleterious that we may now be able to mimic using these new models. So that's what I'm most excited about.
Terrific. That is very hopeful. And I detect the optimism in your demeanor very much. Thank you. That's very exciting. Dr. Rissman, same question for you. What's something happening in dementia research today that makes you the most hopeful?
So I certainly agree with Jim that a lot of our basic science technology and things moving forward is really outstanding. And I think it's going to uncover a lot of new mechanisms for understanding how the disease is progressing. You know, I hate to take an easy one, but I think the most exciting thing is, of course, aducanumab. And the idea that we now have a new treatment that is disease modifying. It also really allows us to fully test the amyloid hypothesis. I think for a very long time, we have thought that amyloid causes Alzheimer's disease. I know a lot of us believe that. I don't think any of us would bet our lives on it. So I think having aducanumab out there and being able to watch it in practice, in people, over a much longer period of time than any one of our trials could, is really exciting. And I look forward to seeing it. And the development of other drugs also in the context of aducanumab.
Great, thank you for that perspective on this week's news. That's great. Dr. Sefji, what would you say? The thing that gives you the most hope right now in dementia research? You're muted.
Sorry about that. You know, again, it's kind of an oversimplification, but I think the thing that gives me the most hope is the number of clinical trials that are coming out, the number of different targets, different types of things. You have people targeting amyloid, people targeting tau, 2 or 3 other mechanisms that I know of that are in clinical trials. So just the plethora and the wealth and the kind of diversity of the things that are being explored is very exciting and gives me a lot of hope.
Great, thank you. This is fun. You all don't know who I'm going to call on next. Dr. Plopper, you're up. What is the thing happening in dementia research right now that gives you the most hope?
Well, the thing that gives me the most hope is the degree of volunteerism out there. I'm just amazed and gratified at the numbers of people who are willing to undergo research, who want to contribute and perhaps benefit, but really, truly, I think there's a real true altruism involved here, and in terms of really wanting to give back to society, and that's what they almost always say. Maybe it's a confluence of baby boomers aging into the very earliest dementia years. That's part of it, I think, because we're seeing just an awful lot of interest, particularly in the San Diego area, in clinical trials in general. So I think that's the most gratifying thing to me is the degree of interest, the willingness of people to become involved, and knock on wood, that the passage of aducanumab is not going to necessarily interfere too much with ongoing clinical trials. We're doing that trial with donanemab. We're also doing the trial with, as mentioned, with lecanemab. So far, when I ask people about that, they generally, they're a pretty sophisticated group in San Diego in terms of knowledge base and being aware of things. And it's not inhibiting them. They want to continue with what the trials. They don't see that as a barrier really for involvement.
Great, I will second that. We have a very knowledgeable, savvy community here in San Diego. Thank you. That is reason for hope. Dr. Mosbach, what's happening in dementia research right now that's making you feel the most hopeful?
Well, if I'm looking at it from the caregiver side, I think for me, what's giving me the most hope in terms of finding solutions, not just for caregivers, but for the individuals who are dealing with the actual disease, is that now the federal government is putting aside money specifically dedicated for only research on Alzheimer's disease or related dementias. And this is attracting a lot of individuals to move into this area of research and try innovative kinds of things. Now, from the caregiver side of things, what I'm seeing with some of that money as well is that there's been a lot of collaborations now with technology-based organizations where individuals who have expertise in artificial intelligence and machine learning are seeking to develop products for caregivers to use in their homes to help them monitor the status of their patients, either through knowing, using geofencing to know whether they've crossed boundaries and maybe at threat to wandering, tracking their behaviors and making sure that the person, and using machine learning to predict issues like behavioral disruptions or agitation, as we were talking about earlier, to help the caregiver know there may be a potential for agitated behavior based on these and these and these factors. So really exciting kinds of new innovative areas to support, to use technology to support caregivers in the ways they most need. I've even seen research applications, and I can't talk too much about it, where even assisted living facilities are getting on board to reassure caregivers that their loved ones are engaging and involved in various kinds of activities in the home environment and socializing, really. So I'm really excited and hopeful that some of these products will make it to market and they'll be available to caregivers soon.
Great. Yes, I hope you can speak more freely about that in the near future. That does sound really exciting. Thank you. Dr. Asen, how about you? What's bringing you the most hope right now?
Yeah, so I think that the last couple of years have brought advances in diagnosis and treatment that are truly amazing. And it means that we can use blood tests now to screen people every year for changes in terms of plaques and tangles in brain that allow us to monitor for Alzheimer's disease many years before the onset of symptoms. And we now have the first approved disease slowing therapy that removes the amyloid from brain, and much progress in attacking tangles as well. It's a whole new age. So essentially, I would say this is the birth of the new age of Alzheimer's diagnosis and therapy, and it's just tremendously exciting.
Well, That is exciting. Thank you very much. Pretty inspirational. Dr. Chun, you get to wrap us up here. What is making you most hopeful in dementia research right now?
It's an incredible universe of possibilities, and there are, what you might view as the traditional explanations that you heard from Paul, but there's a whole nother universe of things that are either not or just partially understood, one of which are things called reverse transcriptases in our brain, which turn out to be fundamental to how our species has evolved. And it's also fundamental to the brain's functions. And there are hundreds of thousands of potential genes that transcribe these types of things in our brain. They're not understood. And if indeed these do have functions in a number of the neurodegenerative diseases, the dementias, then they are accessible by both potentially current reverse transcriptase inhibitors as well as future more specific types of inhibitors. And so this is something that I think is accessible, including through the use of known drugs that have nearly 30 or over 30 years of safety. And so these types of things would be wonderful to look at by the trialist community. Unfortunately, many of these are actually off patent, so there's not an impetus for companies to pay and compensate folks for doing the trials. But I think it's something that should be looked at when we think about out-of-the-box types of approaches. And certainly understanding the fundamental biology will help us understand lots of things about brain, including dementia.
Great. Thank you. Some very, very important points there. Well, this has just been so exciting and inspiring for me. I thank all of you on this panel for everybody in the room here for sharing your Saturday morning with us. Spending Saturday on Zoom is noble endeavor indeed. So thank you all very much for sharing your expertise and your time with all of us. I know we have not answered every question that you had. Please know that we remain available to you at Alzheimer's San Diego. Please reach out to us for information, to get to ask questions. We can connect you to resources for finding more information. I thank everyone for coming. I thank you, Mrs. Shiley, for making all of this possible. I thank Jean and our volunteers who've been here behind the scenes making all of this happen today, and you too, Annie, for that matter. And I thank Eugenia Welch, our CEO, for her leadership of this very fine organization that I'm proud to be a part of. I hope everyone is leaving here today with more knowledge and a lot more hope about where we are in the search for a cure. So with that, I thank you, and we will close the webinar.